Welcome to MOOC course on Introduction to Proteogenomics. In the last few lectures, the eminent speakers Dr. Kelly Ruggles and Bing Zhang talked about genomics, gene mutations and its effects on phenotype. So, now we have better understanding of the importance of mutations in a clinical study and level of impact it can make in the efficient treatment. Therefore, now you know the next logical way is how to do these experiments using the latest technology available, especially how to use various type of NGS instruments to do these kind of experiments yourself. In this light, today we have invited one of the industry leader, Dr. Atima Agrawal from Thermo Fisher Scientific, and she is going to talk about how one could use the next generation sequencing technology and sequence targeted genes in the genome. In today's lecture, she will talk about NGS technology evolution and development over the time, and then there will be hands on session which we are also going to show how to use these instruments for generating data. So, let us welcome Dr. Agrawal to talk to us about recent advancements in the field of NGS and data analysis with a short demo session.
Basically, we are going to talk about the recent advancements which are happening in sequencing field. Like pre 1990, it was a very steady field wherein people were using radioactive labeled ddNTPs, and that's how things were getting sequenced. And then it was quite some time when more than a decade wherein Sanger sequencing was the only sole sequencing technology wherein people were using fluorescent ddNTPs and they were sequencing various fragments. So, past decade there had been many advancements in terms of sequencing. It is not only Sanger now, there are many technologies which have come up in terms of next generation sequencing, and how they primarily differ from a Sanger sequencing or capillary sequencing which all of us were used to for quite some time was that earlier it was more or less it was that one well was sequencing one fragment. Yeah. So, you could have loaded a 96 well plate or a 384 well plate and you could have got 96 sequences or 384 sequences. Now, within this past decade what has happened is there is a term called massively parallel sequencing which has come up. So, massively parallel sequencing is something wherein you are sequencing millions of fragments at one go. So, that is what you are trying to do, and because you have been able to do that, those millions of sequencing together, now you are trying to solve a lot of bigger problems faster. So, that is how you would know that a human genome for the first time when it was being done on Sanger, it took around 10 to 11 years. Now, things are being done in 3 days. So, that is how the technology has advanced, and likewise even the cost at which you were sequencing a genome earlier and the cost at which you are doing now, that is also tremendously decreased. Coming from Thermo Fisher, I would say I am very proud that I was all these decades my company was associated with these sequencing technologies, and we had been pioneer in this arena. So, I would just like to show you very so this is how our Sanger sequences look, and where you are basically plating either a 96 well plate or a 384 well plate and you generate sequences. Here also we have come up with various advancements wherein sequences are now cartridge based. So, the user only has to put in a cartridge and he is good to go, and he just has his prepared samples. This is what we are here to talk about today, next generation sequencing, wherein you have thousands to billions of reads being produced per run, and the data which you are producing is in terms of MBs and GBs. These sequences can be applied to many different fields like metagenomics, microbial sequencing, RNA-seq, exome sequencing, transcriptome sequencing, targeted sequencing. So, all of that, a basic workflow what is involved is, I'll just briefly explain this slide. So, this is the sequencer which we are here displaying, and this is Ion Gene Studio S5 which has 5 different chips which gives you a versatility from producing anywhere from 2 million reads to 130 million reads. That's a very big, like very important feature, so that like if you have this instrument in your institute, a person who wants to do microbial genome sequencing or who wants to just sequence a metagenome, he can also use the same system, and a person who wants to do a full exome or a transcriptome, he can also use the same system. The other big highlight of our system is that one is it gives versatility, that it gives you a various, like it gives you a huge span in terms of the number of million reads. Another big advantage specifically for oncology is that with oncology a very big problem is in terms of the amount of sample you are dealing with. You are generally dealing with tissue blocks which yield very less amount of DNA or RNA as a starting material. So that ways we have a very robust technology called AmpliSeq technology, and this technology has the capacity to multiplex many hundreds and hundreds of targets at one go and amplify all of them at similar efficiency and then sequence them all of them at similar efficiency. So then you are not losing on, so supposingly you want to study 100 targets out of a tumor which is some very few and you get hardly 10 nanograms of DNA or nucleic acid material. Now you want to study 100 targets. So this AmpliSeq technology is basically making, it's such a robust technology that all these primer pairs which are working in one tube, they are so efficiently being mixed that all these targets are being amplified at a similar efficiency, and that is how you are sequencing them at a similar efficiency. So that is what we have pointed it out here, that this sequencer along with its technology has the lowest DNA or RNA input requirements. Then these are the three systems which Thermo Fisher provides: GeneStudio S5, S5 Plus, S5 Prime, and basically they differ in their compute capabilities. In terms of their throughputs and in terms of the compatibility of chips, they differ very in a very minor way, but other than that you have this 510 to 550 chip which can be used with these systems, and the difference in compute power as a user, what it is giving you is faster run times. So a user, a very new user is good probably with S5, wherein run times really do not matter to him much. An established user might would like to go for an S5 Plus who has some planned experiments one by one, and then he just wants, he does not want to get held with the time of run, and then this is S5 Prime wherein you have back to back runs, and this is the technology which has the capacity to finish a run within as little as even 2.5 hours. So this had been on the in this research arena or clinical diagnostic arena for since around 2010, this technology, and it has around 4,500 publications all over. And since we have been focusing a lot into clinical oncology and all, so maybe for us it is more of application notes and all lately, and certain studies which have been done and published in different formats other than scientific publications. So the workflow is what we want to show here is, so a typical NGS workflow would start from nucleic acid extraction. So this nucleic acid can be a DNA, can be an RNA. So once you have extracted this nucleic acid, then you prepare library. Now library preparation is nothing, whatever DNA or RNA you have extracted. Basically it depends on what kind of application you are working on. If you are working on microbial sequencing, then probably your starting sample is a viral DNA or RNA or a bacterial DNA and RNA, and you want to sequence that at one go. So what you are going to do in that library preparation is that you are going to share this genome either by enzymatic sharing or by mechanical sharing. You break down this genome into small pieces and then you ligate adapters. These are double stranded fragments which are ligated on both the ends, and that is what you do. Like basically what you are trying to do is you are sharing down the interested genome into smaller parts and ligating it with adapters. The sequence of these adapters is known, so that is how the primer can sit and it can sequence the region in between. So, that is what you do in a library preparation. Now, for an oncology sample wherein you are trying to look for different markers specific for a particular cancer or particular type of cancer, what you are going to do is you isolate DNA and RNA, and we have wonderful panels which are like spanning from we have panels for solid tumor, we have targeted panels for heme-oncology, we have panels for cell-free DNA or RNA, that is liquid biopsy, which is gaining a lot of attention these days. Then we have panels for addressing the immuno-oncology also. So, you use any of these panels, you amplify the targeted portions, and then once you have prepared this library, what you do is, this is your template preparation. During this part, what you are doing, you are amplifying these library molecules onto the ion spheres. This is being done onto this system wherein what you are doing is these are two tubes, wherein in one tube there is oil and in one tube there is a recovery solution or some kind of a detergent. This you place your plate here and you place a filter wherein your sample is kept with the ion spheres. This passes through this instrument and goes onto this plate. This plate is Peltier controlled, yeah, on both the sides, and that is how all these library molecules are then getting amplified onto the ion spheres, and from here, from on this tube, they are passing into a centrifuge which has two tubes. So, all these amplified ion spheres are getting passed onto these two tubes, and finally you are taking your amplified ion spheres out of this. Then you have an enricher. We have subsequent slides wherein I can tell you the biology of it. You have an enricher wherein you have an A12 strip kind of a thing placed, and what you do is that your amplified ion spheres, because probably not all of the ion spheres have amplified. So, what you are doing is that you are placing that mixture here. When you were amplifying, you were using a biotinylated primer. So, whatever has amplified, whatever ion spheres have amplified, they have now biotin label on them. So, what you do, you place the streptavidin coated beads in one of the well, and this is again a semi-automated way. This system, this is called enrichment system. This will go on its own for half an hour, and then finally through this biotin streptavidin binding, it will fish out all the amplified ion spheres and it will leave the non-amplified ion spheres, and once that is done, I will just cover that part in the slides first. So, we have two solutions. One is the semi-automated way which we have presented here. We can show you the videos for the fully automated way of sequencing, wherein you use an iron chef. So, wherein you do not have to deal with one touch 2 and enrichment. What you do is this iron chef, this serves two purposes. One is it can prepare libraries for you, and second is it can do all the amplification on these ion spheres, and you get ready chips which are already being loaded. These chips, these are basically silicon wafers on which there are millions of wells. These millions of wells are basically enabling these millions of fragments to be sequenced all at once. So, what happens is on the system, so this is the, once you have your sample prepared, if you are using iron chef, then your sample is automatically loaded onto these chips. If you are using the semi-automated system, then you have to load the sample with a pipette, and once it is loaded, this is the sequencer which takes not even 15 minutes to set it up. So, there is a lot of apprehension that a next generation sequencer is very hard to kind of handle, and it's difficult to manage that kind of a system. That is what we want to show you, that this is the chip clamp wherein the chip goes and sits. These are already bottled reagents, wherein if you see, this is a sequencing cartridge wherein all the 4 DNTPs are being placed. Then behind this there is the waste cartridge. This is the wash solution which goes onto the chip every time sequence polymerization has happened, and this is the solution, this is the wash solution after every sequencing run. This wash solution goes onto into the fluid lines, and that's how it cleans the system. So now you can imagine how much time it would actually take you to pull these cartridges out of a aluminum foil and place it into the instrument. The instrument on itself, once you have placed all these reagents on the instrument, so the instrument takes around 40 minutes to initialize itself, and then you already have your loaded chip here and you are good to go. And this is the fastest system which can produce BAM5, which can produce data or which can finish a sequencing run within as little as 2.5 hours. So I had shown you various models of the chip, starting from 510 chip to 550 chip. These chips have different capacities. For an ion chemistry, you can, in terms of read lengths, you can go from a 200 base pair to 600 base pair. Yeah. So why these read lengths are important? You consider different read lengths for different kinds of experiments. So supposingly your intention is to sequence certain markers from a tumor sample. So now a tumor biopsy, as it is, whatever DNA or RNA if it's not a fresh frozen biopsy, it's something like a FFP, it's coming from an FFP block, that DNA or RNA itself is highly degraded. So then you will use a smaller sequencing chemistry to sequence these small portions. If your intention is to sequence a metagenome, then you are trying to deduce various kinds of bacteria or viruses present in that sample, whether it is a sample which is coming from a cancer biopsy, whether it is a sample coming from an environment, whether it is a sample coming from a soil. So if you are studying metagenomics, you would like to read as long as possible, then probably you would go in for a 400 or a 600 base pair chemistry. So that is how things differ in terms of read length. And what I want to point out is, my colleague Harsh is going to talk about the data analysis part very fast. So we are not only leaving you at the sequencing part, we have full fledged solutions in terms of data analysis, which takes you, and further for clinical people, it also helps in generating reports. So reports which can be directly given out to patients and be told that, okay, this is the therapy which you could be using for treatment. So going to the next slide, these are just some figures of the workflow. So this is the library preparation what I was talking. So supposingly I am just taking an example of one of the assay. This is Oncomine focus assay, which has 52 genes. These are all oncogenes, and in the form of hotspot mutations, CNV gains, fusion drivers. So it is a very well known fact that if you are working with oncology and if you are working with cancer genomics, you could not be only looking at single nucleotide variations, or you could not be only looking at insertions and deletions. You have to look, if you are looking at genomic aberrations, you have to look them in totality. So you have single nucleotide variations, you have CNV gains or losses, you have fusion drivers, everything. Now if these are the kind of mutations you are looking at, your starting sample has to be RNA as well as DNA, because these two parts of the markers are going to come from the DNA, and fusion drivers are going to be reported from the RNA. So what you do is you isolate DNA and RNA, you reverse transcribe the RNA, and then you have this pool of DNA or cDNA. You take a primer pool. Now this is what is the primer pool coming from. So Oncomine focus assay, which is dealing with 52 genes. So this primer pool will be targeting all these 52, all the hotspots and CNVs and fusion drivers in these 52 genes, and this is going to be one vial. And your sample, which is anywhere from even as less as 1 nanogram, goes and into this one vial, and all these targets, so these are basically 52 genes which are actually addressing as they are addressing around 1000 biomarkers. So all these 1000 biomarkers are getting amplified in a single tube, and so this is that amplification happening. Once you have generated these amplicons, because these primers are coming from conserved regions, you partially digest these and then you ligate adapters. This is what we discussed about library preparation, that you want to ligate the sequence of interest to adapters wherein the sequence is known, so that the primers can come and sit here and sequence the region in between. So, and we can use barcoded adapters. Why we are using barcoded adapters? So that we can use more than one sample on a chip. So, you have seen the chips, and so basically you would like to sequence more and more sample. So, it would depend. We already tell you that okay, on this particular chip for this particular panel you can, how many samples you can multiplex, that comes with our literature. So, that is how you are using these barcoded libraries. So, this was the second step, which is template preparation, which can be done on a semi automated way. You would use an OT2 and an enricher. When you want to opt for a fully automated way, it is Iron Chef, which we could not get here, but we have a video, we will show you that video. So, what is primarily happening is these are your hundreds and thousands of library molecules which are coming from the amplified portion which you have amplified from the sample, and then you the idea here during template preparation is that this one library molecule gets clonally amplified onto the ionosphere, so that when this molecule is being read, the signal is enough that it can differentiate noise and the signal. This is what is happening. So, this is an ionosphere which has an oligo which is complementary to one of the adapters of the which are linked to the library. So, this is your library molecule which on both sides is linked with an adapter, and this is the primer, and the primer extends. So, this reaction is on this plate and this is Peltier control. So, all these small droplets which are being formed during the emulsion PCR, they are working as individual PCRs. So, these library molecules are then getting amplified, and what finally you are getting is you are getting this particular library molecule all over amplified onto the ionosphere. Now, you are using streptavidin coated beads on this system, and that is how you are fishing out the amplified beads with non-amplified beads. And this is the Iron Chef system, which is a fully automated way into how you prepare your libraries. So, when you are preparing libraries using Iron Chef system, you prepare your DNA samples and you load them into these cartridges, and there are various other cartridges which are put in different different sections of the system, and that is how the final you get a final library pool into one of these tubes. Right now what is being shown is the template preparation step on Iron Chef. You again load these cartridges, and then the system does what is being done on this plate. It is being done in a 96 well plate here. The emulsification and the amplification of that emulsion is happening, and then enrichment is happening through this cartridge, and finally this is the centrifuge wherein you have your chips loaded. So, the chips are also loaded automatically, and then these chips are ready to go directly onto the sequencer. So, this is what we had seen. So, in the sequencer you have first the chip which goes into this clamp, which is a temperature controlled clamp, and you use polymerase, and this polymerase, because it works at a specific temperature, that is why this clamp is temperature controlled. So, you already have your sample loaded onto this chip. Then this is the sequencing cartridge which has all the 4 nucleotides DNTPs which are flown onto this chip in a sequential manner. This is the wash solution, this is the discard which goes at the back of the wash solution. This is the wash solution which goes onto every time there is a nucleotide being sent to on the chip, then this wash solution goes and it washes any extra DNTPs left on the chip. And this is that wash solution which is placed here, which you can see, which cleans the system automatically after every run. So, it practically does not take a user to more than 15 minutes to set this sequencer up and get going with the run. So, that is all. So, I practically shown you, you install the waste container, wash solution, cleaning solution and reagent cartridge, you install the chip and you select the run plan and you are good to go. This system also does not need any standalone computer with it. It can be connected with LAN and then you can operate on the system from anywhere. So, what is happening in terms of sequencing? So, as you had seen, this chip has millions of wells And in each well what is happening is, now this is supposingly an iron sphere which has amplified molecules onto that. We have already added primer which is against the adapter. Then the sequencer, what it is doing through this cartridge, it is pumping various DNTPs in a very sequential manner, which is already known to the algorithm of the software. So, what happens is, the moment there is a DNTP flown in onto the chip, so supposingly we are flowing in an ATP as the first nucleotide. So, now this particular action is taking place on millions of wells on the chip. So, all those wells which have an iron sphere which has a T as the first base, and this ATP will go and bind. The moment there is a phosphodiester bond being formed, there is a hydrogen ion release. This, and because we did clonal amplification, we amplified one library molecule all over the iron sphere. So, there is not one hydrogen ion release, there are going to be thousands of hydrogen ion release which are good enough to bring a change in pH for this particular well. Each well has its own sensor. So, now the system, now the software will calculate, or while the run is going on, the software algorithm is also going on, and it is seeing that from the first phase, from the first run of DNTP, when that was flown onto the system, how many wells showed a polymerization, and in how many there was a change in pH which got translated into a change in voltage, and that is how we get a signal. This signal is very well differentiated from noise, and that is what gives you the accuracy in data. So, what is happening is, the nucleotides are flowing sequentially over a chip. There is one sensor per well, there is direct detection of natural DNA extension, and then this phenomena is happening in millions of wells at one time. So, just a cartoon to show this in a better way. This is your ISP. These are those two adapters which you have ligated during the library preparation process, and this is the sequence in between which you want to sequence. So, what is happening is that the primer which we have added, which the system has already added before the sample is being loaded onto the chip, the primer comes in a needle's hair. There is a sequential addition of DNTPs, and so a T will come and bind. There would be a change in pH which will be converted into change in voltage, and that would be read as a signal. Then again, now what happened was, now this system has flown in a DATP. Now the second base in question is G, because they do not have complementarity, this particular base will get washed off. Then this sequential addition of DNTPs will go on, and signals will be captured. What do you think will happen at this stage? There are two A's and we are flowing in a T. Signal will be double, and that is how the system will know that there were two A's. Yeah? So, the signal will be exponential if there had been 3 A's. So, that is how it will work, and based on these signals you get finally a sequence, which is then a sequence coming from that particular well out of those millions of wells. So, after a 2.5 hour run, you have probably 80 or 100 million such sequences which now need to be analyzed. So, this is how a typical run report looks, that this is your chip, this is the area wherein you have loaded the sample, and this I will not go into these parameters, and this red, basically this is the gradation in terms of how good the chip was loaded. This is 90% loading, and finally it will give you some statistics. Now this is a 530 chip run, and what we have done is we have run a liquid biopsy panel for lung, which is targeting the variations which are there in DNA. So, that will include single nucleotide variations and insertions and deletions, and this is the typical read length, because so as you would all know that liquid biopsy is gaining a lot of attention because of its non-invasive nature, because it gives the way that it is being sampled, it gives a better representation of the tumor, because if you are taking a solid tumor, it could, we all know its tumor sample is heterogeneous. So, what sample we are actually taking the DNA out, whether that is representing the actual tumor position, tumor condition, we do not know. So, liquid biopsy that way is a better way of handling, and more so because it is a non-invasive way. So, it becomes a very good prognostic marker. So, especially when you are dealing with lung cancer cases, you are not allowed to do biopsy many times. So, after a point of time, or at times there would be patients wherein you are just not having that flexibility of taking any biopsy. So, there these liquid biopsy solutions are coming very handy. This again is a panel which has some 22 genes or things like, and these are all multiplexed in one tube, and they are all getting amplified at one go. So, this is, so you generated so many million reads, and these are the read lengths, because it is a cell-free DNA, as it is by nature, it is a fragmented DNA. You do not get DNA fragments more than 120 to 170 base pairs. So, you would like your primer pairs to be amplifying very short regions, so that you do not skip on that.
So, today you have learnt about how sequencing genome or a specific target gene sequencing has now become very easy, accessible and affordable to many of the researchers, which could facilitate them to dig deeper and solve many clinical challenges. After understanding about genomics and looking at some of these hands-on sessions about various technology platforms used from the leading industry leaders from the Thermo Fisher or Illumina Scientific, now we are going to move on to the next module, which is on proteomics. So, in the next module I will talk to you about fundamental concepts of proteomic technologies, and then we will have other eminent speakers who will talk to you about current workflows using mass spectrometry based proteomics and how to do different type of tools and databases for proteomics analysis. So, let us stop here for the genomics module, and of course, you got excited, you have learned lot of new things here, but you have also now got exposed to various type of publicly available databases and repositories which you can download yourself and now start implementing those using various tools available and showed in this workshop, from which now you can address many specific questions, and you can probably do many experiments even without having non-availability of even the technologies or the NGS system in your lab or in your own place. You can still do many of these experiments and obtain some very meaningful information. Thank you.